opportunity, and I don't know if you ended up getting snow up there, but we're certainly getting it here in Ames today. I am an associate professor in agricultural and biosystems engineering at Iowa State University, and really pleased to share some of our work on antibiotic resistance and mitigation. And we've been working on this since I came to Iowa State in 2008, and this is one of the first projects that we started working on. So Amanda asked me to kind of frame this as far as what are some of our biggest questions and what are some of our most exciting results. So I thought I'd start out with the questions. And one that has probably already been touched on a little bit today is how do we monitor resistance in the environment? So we have done some monitoring of the actual antibiotics. There's a lot of challenges with that, and so much of our more recent work has actually been looking at the microbial resistance. And so we do a lot of different things. First of all, we look at the phenotypic resistance. So we'll plate for fecal indicator bacteria, and then in the cases when we know what antibiotics are being used, we'll add those antibiotics to the agar and get a fraction of phenotypic resistance. We've also been doing a lot of qPCR to try and detect certain resistant genes and mobile genetic elements. So a lot of our work has been with the swine industry, the pork board, and so we've been looking for macrolide resistant genes, and also we've been expanding that to some tetracycline resistant genes. And then we've also been collaborating with Dr. Adina Howe, who does bioinformatics and metagenomic sequencing, and so we have been working with that to try and expand beyond these kind of key genes that we've been working with to look at the manure-derived or the resistance community. So how can we track the resistance in the environment, especially when we try to scale up beyond the farm and we have different facilities with different antibiotic use patterns? How do we really track what that resistance looks like at a larger scale? Some of the other questions that we have, which I don't necessarily have an answer to, is how is the VFD going to impact environmental resistance? So we've got some pre-VFD data, and it's just going to be really interesting to see if we see an impact fairly quickly in reduced resistance, or if it's more of a lag time. I think of phosphorus in the environment with this a legacy effect, and what kind of legacy effect might we see with AMR? And then what is the role of environmental AMR in the human health picture? I think that's something that we'd all like to have a better sense of. So key points for AMR mitigation. We are currently working on the manure management aspect. So what are some ways that we can treat the manure before we apply it to the land? So can we mitigate prior to land application? We've been studying in-field management, so different methods of applying manure application timing, and then also we have some new work starting on edge-of-field management, specifically with prairie strips. So I'm going to talk through the results of some of these studies. So this is our manure mitigation experiments, which have been funded by the Pork Board. And we're considering a lot of different options to see what kind of reductions we can see in the phenotypic resistance and then resistant genes. The gene that we are working with is ERM-B, and so that's a macrolide resistant gene. So our manure is coming from the farm where tylosin is used. Up at the top, you see I have ionophore addition and solid separation. We tested the ionophore addition just because it's used in the pit foaming. Barns that have pit foaming problems will add the ionophores to try and reduce the methanogen populations, and so we wanted to see if that had an overall impact. So far, it doesn't seem to have any impact on the resistance community, and then solid separation is still pending. The first figure that I have here is showing the two-phase storage. So if you look at the left, we have no fresh manure added, and on the right is the scenario where fresh manure is being added on a continuous basis. So the no fresh manure added would be a situation where there is a pit underneath the facility and then there is secondary storage. And that would allow the manure to sit for about 6 months before it would be land applied. And in this case, you can see that the y-axis is the ERM-B copy numbers, and then the x-axis is the time since the start of the experiment. And when no fresh manure was added, it looked like in the ERM-B populations we were starting to see a decline over this time period, except for that last data point. But then when you look at the situation where fresh manure is continually being added to the system, we continue to see an increase in our ERM-B copies. Next, we looked at anaerobic digestion. We considered 3 different temperatures, and the results are the days for a 1 log reduction of tylosin and tetracycline resistant Enterococcus. And so just preliminary, you can see that there was, at the higher temperatures, a shorter period for that 1 log reduction to occur than at the 25 degrees Celsius. The third option that we considered was centrifugation. And this is just looking at the Enterococcus tylosin resistance over a range of different centrifuge times and centrifuge forces. And you can see that over, either a longer period of time or a higher gravity force that we were able to see reductions in the resistant populations. Okay, so this is still all lab scale stuff, but we worked with Dr. Anderson, who does manure management here at Iowa State, to identify practices that would be feasible for farmers, and ongoing results will be expanding the qPCR and then to do more of the sequencing. So next slide is looking at some of our in-field management studies. And these studies have been conducted at the Iowa State Northeast research farms, and essentially 1 acre plots that have either received manure or a no manure control, and they have a long history, over 30 years of no manure. And then we monitor tile drainage systems. So we're really looking at the subsurface transport of these resistant genes. And the research question is looking at both the crop rotation, so we've looked at a corn and soybean rotation. More recently, we're looking at corn on corn, and how do those resistant genes move to drainage water. From this, we can see that in the soil, the resistant gene levels return to that baseline concentration within about 6 months to a year after manure application. So if manure application occurs in the fall when it's dry conditions, in some cases, that's kind of the climate trend has been in the past, has been dry conditions in the fall, and especially if that manure application can be applied late in the year prior to when soil temperatures are dropping but before freezing. And then there's a lot of mitigation that can take place in that soil and reduction in the resistant gene concentrations before spring, when water starts to move through the system and we start to see stuff moving off-site. We did find that plots receiving manure had greater concentrations of ERM, B, and F, so 2 macrolide resistant genes. And also we're demonstrated that the fecal indicator bacteria do not correlate with these ERM genes. So the FIBs that we've used for many, many years to detect the risk to human health really aren't correlating with any of the resistant gene concentrations that we've been studying. So this is a prairie strip. So this is new work funded by USDA to look at prairie strips as a mitigation strategy, so an edge-of-field mitigation strategy for AMR. And then this would be really focused on the surface pathways. So the overland flow, the water would flow into the prairie strip where the resistance community could be intercepted, pushed into the soil, because prairie strips will increase infiltration. And then we also know that the microbial community in the prairie strip is different than an a soil that's under corn and soybean or corn and corn rotation. And so we are wanting to test to see what kind of persistence occurs both in the ag soil and then within the prairie strip community. So the picture there is of some rainfall simulation experiments that we conducted this past fall. And then the figure is showing essentially differences in the microbial community across a gradient starting in the field and then moving into the prairie strip. So you can see that the The green section, that's the microbial community in the corn and the soil that was planted to corn. There's kind of a stripped corn interface, and then as you move deeper into the prairie strip, you could see that the community shifted, and we could see differences there. There was also an old fence line that was, clearly had not been on ag production for many years, and so we pulled some samples from that site, and that community was also completely different than the corn and the prairie strip. So a lot more work in this area over the next couple of years. And then the last project that I wanted to share with you is some of the watershed scale monitoring. So you can see the map of the watershed. That's the South Fork of the Iowa River. It's a large watershed, and each yellow dot is a location of a CAFO. And so this is a really heavily manured watershed, and we partnered with USDA ARS. They have an intensive monitoring network in this watershed, and we were able to get drainage and river water over several years and from multiple locations within the watershed. And so if you look at the table on your far left, there's 3 surface locations, so those were samples that were collected essentially from the river, and then 2 locations that were from tile drains, and then the drainage area that corresponded to each of those catchments or sub-watersheds, the number of samples. And then what I wanted to point out is the 2 for ERM-B and ERM-F, the limit, the percentage of samples that were above the limit of quantification. And we always saw greater presence of resistant genes in the drainage samples than we did in the surface sites. And so this has been, it's been really interesting to see that. It also, as we've talked a little bit about today, I think emphasizes the role of the soil in resistance. If there's a mobility and gene sharing and gene transfer that's taking place, the soil environment is a place where that could happen. And that's possibly reflected here in these results as we see these higher levels of resistant genes coming through tile lines than surface. With almost any other pollutant, you would expect to see higher concentrations in surface runoff, so in a surface-driven system than in a subsurface system. Usually you see a lot of filtration that takes place as the water moves through the soil profile, and except for nitrate, of course. But in this case, it was very interesting. Other studies in the same watershed have looked at E. coli, so just the presence of E. coli in the watershed, and they're seeing greater concentrations in the surface sites than in the tile. So more work is looking at this watershed. We've sequenced the samples now and are looking at the 16S and the microbial communities among the surface and the tile drainage points. That's all I had. Just wanted to make sure and acknowledge the collaborators that we work with, Dr. Adina Howe, Dan Anderson, Tom Mormon, and Heather Allen. Tom and Heather are both with the USDA ARS labs here in Ames. We've had grad students, and then funding has come from the USDA Food Safety Grant and a couple other USDA grants, as well as the National Pork Board.